Hello everyone, and welcome to our presentation entitled Evaluating and Managing Nutrient Inputs from On-Site Wastewater Systems in the Falls Lake Watershed, a multi-scale approach. In this presentation, I'll first start by talking about a background on on-site wastewater treatment systems or septic systems. We'll then discuss how you can manage septic-derived nutrients using natural and nature-based features. I'll show you some estimates for the number of septic systems in the Falls Lake watershed. Then from there, we'll transition into discussions on the funded studies. I'll show you the research questions these studies sought to address. I'll show you the results from the NCDEQ 319 funded study and also the NC Policy Collaboratory study. Then I'll summarize and talk about future steps. So to give a bit of background, in North Carolina, excessive chlorophyll A and nutrients are among the leading causes of lake impairment. In fact, septic systems are commonly cited as an important non-point source of nutrients to surface water. However, there are limited quantitative studies that have estimates of septic-derived nutrient inputs to surface waters, especially at the watershed scale. I think this is due in part to how challenging it is to generate accurate estimates of septic nutrient inputs to watersheds. And I've listed some of these reasons here as to why it's so challenging. One being that wastewater effluent that's discharged from systems is diffuse. Eventually it'll mix in with groundwater systems and travel with the groundwater, and it becomes challenging to track these plumes when you start talking about multiple systems within a watershed. There's also a lack of septic monitoring data, both from the context of water quality data, but also from the context of GIS data. Just knowing where septic systems are located can be very helpful in estimating some of these watershed scale nutrient loads. And there's also just the complexity of nutrient transport. So nitrogen and phosphorus can undergo many different transformations throughout its flow path. So if septic systems are a potentially significant nutrient source to surface waters, then one important question becomes how can we actually manage these septic-derived nutrients? And natural and nature-based features offer a potential solution. So take, for example, if we have a septic system with a contaminant plume, which you can see here, if this contaminant plume happens to upwell in an area where you have a buffer present, and if that buffer is healthy and has an extensive root zone that can actually reach the water table and presumably the contaminant plume within the groundwater system, it's possible that you might start to see denitrification and immobilization within that plume, therefore reducing the amount of nutrients before it reaches the stream. Now, if this buffer is maybe absent or unhealthy, or maybe the water table is deeper than the root zone will reach, and then there are other engineered solutions like subsurface denitrifying bioreactors, which you can see here, stream adjacent bioreactors here, or an in-stream bioreactor, which typically contain carbon-rich media designed to facilitate denitrification. And in some cases, adsorption and precipitation of phosphorus can also be plausible. So if we think about the amount of septic systems in the Falls Lake watershed, there are about 50,000 septic systems based on estimates by Brown and Caldwell, and most of those 50,000 occur as conventional functioning subsurface discharge systems, about 44,400 of those. If we look at this map on the left, this is a map of the Falls Lake watershed, and everywhere you see a purple circle or a red circle, that's indicative of a state permitted or a county reported septic system. So these are known and suspected locations of septic systems, and the map on the right is just a little bit a simpler way to show the density. So it's a density surface based on these points, and everywhere you see those hotter red, darker red colors, that indicates a higher septic system density. The green polygons represent septic watersheds that we were actually monitoring, and the purple polygons represent the sewer watersheds that we were monitoring. So this takes us to our research questions, and more specifically, we wanted to address how does nutrient attenuation vary at the system and landscape scale? Do septic dominant watersheds contain elevated nutrient exports relative to sewer watersheds? Is there a difference based on geologic setting in terms of nutrient loading from septic systems in the Falls Lake watershed? And then also, what bioreactor porous media are most effective at reducing septic-derived nutrients? So to address the first research question, we identified 5 sites that are all residential sites served by septic systems. 4 of these sites are served by conventional gravity draining septic systems, and 1 was a sand filter site. All these sites are located in Triassic Basin geological settings. More specifically, they drain to the Lick Creek subwatershed, which is a subwatershed of the Falls Lake watershed. You can see sites are located right about here, and they're all within the same subdivision. So we identified these sites so that we could quantify nutrient treatment at the individual system and lot scale, and using these data we could then calculate wastewater nutrient attenuation at the system and landscape scale. So jumping right into the data here, just to point out the color scheme here, the purple denotes the septic tank or the wastewater concentrations or nitrogen chloride ratios. The gold is the groundwater beneath drain fields. The green are groundwater that's down gradient of the drain fields. The light blue is a sampling port in a septic bed system, and then the light pink is the sand filter effluent itself. So if we look real quickly across all these sites, we see this trend of decreasing, right, between the tank and wells down gradient of the, or features down gradient of the tank. And what we see with regards to concentration reductions, they ranged from about 64% to about 95% across all sites between the tank and the groundwater beneath the drain field. So some pretty decent reductions there, though there are some sites that still had elevated TDN concentrations. And as we got further away from these septic systems, we noticed generally an improvement in the nitrogen concentration reductions, ranging from about 80 to 97%. Now, we also wanted to look at nitrogen chloride ratios so that we could figure out to what effect dilution might be playing, since concentrations can be diluted, and get an estimate of the mass reduction. And by and large, we see a very similar trend, declining nitrogen chloride ratios, which suggests that nitrogen is being removed through denitrification or immobilization. And these mass reduction percentages range from about 18 to 93% between the tank and the drain field, and about 53 to 74% between the tank and down gradient piezometers. So switching gears a little bit, we're going to be talking about the sub-watershed scale monitoring efforts. So for these efforts, we needed to select sub-watersheds based on wastewater treatment approach, septic or sewer, and also geologic setting. So we located 28 sub-watersheds for the DEQ funded portion of the project, which included 22 septic watersheds and 6 sewer watersheds spread throughout the Falls Lake Basin here. There are few in the Triassic Basin, which we have data from past studies on, so we wanted to focus more on the Carolina terrain and also the Falls Lake and Crabtree terrain. Now for the NC Policy Collaboratory study, we identified an additional 15 sub-watersheds that were served by sewer, most of which were in this kind of Wake County region. The density of septic systems in these watersheds ranged, but the maximum was approximately 2.5 systems per hectare. And for this presentation, just be focusing on the nitrogen and phosphorus or phosphate, just in the interest of time. But we did collect quite a bit of other data and parameters, logged some data, and did manage to get a storm sample as well. Moving on to some of the key results here, what we found was that septic watersheds contained elevated concentrations of TDN, nitrate, and phosphate, and these differences were significant. Now, when we think about, okay, this is the watershed scale, how do we know these nutrients are from septic systems? So we also took isotopic data or samples for isotopic fractionation, and what these data suggested was that wastewater was a dominant source of nitrate, not just in the septic watersheds, but there were also some wastewater signatures popping up in the sewer watersheds as well. So when we reclassify these subwatersheds by geological setting, we see a little bit of a different trend here. We still see significant differences between septic and sewer when comparing the Triassic Basin septic watersheds to the Triassic Basin sewer watersheds. Again, elevated concentrations of TDN, nitrate, and phosphate, which are significantly different. But when you look at the other geological settings, the Carolina terrain, the Falls Lake and Crabtree terrain, we don't see significant differences. The medians sometimes are a little bit elevated, but those differences, again, are not significant. So thus far, we've seen that areas with septic systems contained elevated nutrient concentrations at the lot and watershed scales. So the next question becomes, how can we effectively manage these elevated nutrients? Which leads us to our final question, where we wanted to consider what types of carbon-rich media would be most effective at removing nitrate. So in this project, we had 9 pilot-scale bioreactors, 3 using peanut hulls, 3 using pine bark, and 3 using wood chips. And each of these bioreactors were dosed with 20 milligrams per liter of nitrate at varying hydraulic retention times during 3-week-long trials that began in June 2021 and extended through November 2021. So we found that all of the carbon-rich media were effective nitrate removers, though there was some difference in overall treatment. Pine bark contained the highest nitrate removal percentage of about 76% and was significantly different from peanut hulls at 70% and wood chips at 65%. To summarize, we found that septic systems can be significant sources of nutrients to impaired and/or nutrient-sensitive waters, especially in areas where geologic, soil, weather, or other characteristics negatively affected system or landscape's attenuation processes, or when septic system density is elevated greater than about a system per hectare. We also found that watersheds served by septic systems in Triassic Basin settings contained elevated nutrients relative to all other watersheds, which suggests that other high-density septic watersheds in Triassic Basin settings may also contain elevated nutrients. And finally, we found that pine bark, peanut hulls, and wood chips were all effective carbon media at facilitating nitrate removal in pilot experiments at various hydraulic retention times, which suggests that these technologies could be utilized in the form of in-stream, stream-adjacent, or subsurface bioreactors to help improve nutrient attenuation in areas with elevated densities of septic systems. The last thing I'd like to discuss is the future steps moving forward. In the current studies, we focused our monitoring efforts at the system and landscape scale in the Triassic Basin. Thus, adding more research sites in other geological settings is important to help further constrain nutrient attenuation modeling efforts for Falls Lake. Additionally, continuing to monitor subwatershed and watershed scale nutrient transport, especially in areas with varying densities of septic systems, is important as these data can help further understand watershed scale attenuation factors. And finally, in areas where we suspect or know there are elevated concentrations of nutrients, adopting best management practices may be a beneficial or effective way to reduce some of the nutrient loading to Falls Lake or its major tributaries. Thank you for your attention, and if you have any questions, I'll be happy to take those during the Q&A session.